Hello everyone, welcome back to the second installment on the immune system chapter series. I'd like to start by introducing everyone to the new section. So in the second part of this chapter we'll be talking about adaptive immunity instead of innate immunity and the ways that receptors in adaptive immunity can provide pathogen-specific recognition. So as we had discussed before, invertebrates only had innate immunity, but now we're moving on to vertebrates who have both innate and adaptive immunity. And so the adaptive immunity part of the invertebrate system relies on the use of T cells and B cells, which are both types of white blood cells called lymphocytes. So the book describes the lymphocyte literally as a white blood cell that mediates immune response and consists of T cells and B cells. So these lymphocytes originate from stem cells and bone marrow, and some of them migrate to the thymus, which is an organ in the thoracic cavity right above your heart. And the lymphocytes that mature there are called T cells, T for thymus. And the lymphocytes that stay in the bone marrow develop as B cells, B for bone. And then lymphocytes that stay in the blood become the natural killer cells that we talked about in the last section of the chapter. And so foreign things that come in that elicit response from the T or B cell are called antigens. And in adaptive immunity, it occurs when the T or B cell binds to an antigen, such as bacteria, virus, and they bind via the antigen receptor. The book goes on to say that all of the antigen receptors made by a single cell, B or T, are identical, and when an infection occurs, it triggers the activation for a specific part of the pathogen that is entered. And it also says that it's noteworthy to know that the cells actually contain about 100,000 antigen receptors on the surface. You can see right there. The antigens are usually foreign and very large molecules, and many of them protrude from the surface. And as you can see down here, the part of the pathogen that binds to the T or B cell is called the epitope. Moving on, this section just says that they bind to many different, the pathogens bind to many different cells. Moving on down here, we begin to talk about B cells and how they function and are made up. However, this section really just describes everything we're going to see down here in figure 43.9. It talks about how each B cell has a Y-shaped antibody on it that has a heavy chain and a light chain with these disulfide bridges linking them, and each type of chain has a constant region which varies little between unique types of B cell, not unique, single types of B cells. So between many different B cells in your body, these constant regions aren't going to be very different, but these variable regions are going to be different. The book states that the binding of these antigen receptors to an antigen is what initiates B cell activation, which creates a cell that can secrete the antibodies. And the antibodies, rather than the B cells, are what actually fight the infection in your body. So if we go up here where it actually talks about antibodies, you can see that antibodies are the Y-shaped antigen receptor that comes off of the book describes it as a differentiated B cell. So it's the protein secreted, binds to a particular antigen. So this section just says that, like with most other receptors, antibodies are very good at having a unique shape that provides a lock and key fit, like you will see in the next figure. That and the variable regions that we had talked about provides the high specificity needed for adaptive immunity. Figure 43.10 A and B down here. Part A shows you that the antigen receptor, which is the antibody still connected to the B cell, codes for the same exact lock and key fit as the antigen because, the antibody, because they are the same thing. So they will match the same epitome on the antigen. Now down here, part B shows you the specificity that the variable regions on the antibodies. Antibody A has a different shape than antibody B, than antibody C. So they can code and find a large array of antigens. Moving on again, we are now going to talk about T cells and their antigen recognition. So the T cell antigen receptor, like the B cell, as you can see over here in figure 43.11, will just be used to demonstrate. You have constant regions, variable regions, and the variable regions up here are connected together to bind to the epitome of a pathogen, and the constant regions, which uniquely to a T cell contain a transmembrane region that bind to the plasma membrane of the T cell, are at the base, and you have two chains here. This is new to the T cell. You have an alpha chain and a beta chain, but again, these chains are connected by a disulfide bridge, just like in the B cell. The book also states that while B cells bind to epitomes of intact pathogens, the T cells bind to fragments of antigens. Now, as the book moves on to talk about how T cells begin to recognize things, we move down here to figure 43 point, I think it's 0.12, yep, part A. We've got, like we said, a host cell that has been infected by a pathogen, and some molecule in the host cell has taken up particles from the pathogen. This MHC molecule is a major histocompatibility complex. Takes in a part of this pathogen and protrudes from the cell surface. The T cell antigen receptor comes and binds with this MHC molecule. Now that was just a general overview. This part over here specifies that each of these orange pieces is called an antigen fragment, and you've got this arrow is showing what's called antigen presentation. And when we move down to figure 43.12b, this is just a close-up of what was described as the antigen presentation. This is a top view. You've got this green here is the major histocompatibility complex, and it's hosting this antigen fragment so that the T cell will come in here. Well, the T cell antigen receptor will come in here and bind to this orange presented antigen fragment. Now, for the sake of time, this section just talks about development, and it doesn't give any hard facts, so we're going to move forward and specify more about that later. And really, anyway, it just discusses how well adaptive immunity works because of the specificity and development of the T and B cells. Now, moving on to here, this section discusses we have person makes 1 million different B cell antigens, 10 million different T cells. So how, with only 20,000 protein coding genes, do we have so much specificity? And it talks about combinations, and it exemplifies here an immunoglobulin, which I think I actually skipped over on accident. An immunoglobulin is an antibody. If you see IG, that just means antibody. Immunoglobulin, antibody are the same thing. Now, this whole section up here once again, we're going to go down to the figure for visualization, and I'm just going to go through things. So figure 43.13. So we're talking about, we have a single undifferentiated light chain B cell gene so an immunoglobulin gene, and we have various variable segments. So these are arbitrary: variable 37, variable 38, 39, 40. And we have J sections, which up here you can see they're called joining segments. And so we have 5 of those, an intron, and a constant section. So the combination that's gonna happen, and how we get so many different varieties from a single starting point, is an enzyme complex called recombinase is going to come in here, and you can sort of think of everything that it removes as an intron, and the intron is going to be removed for RNA processing. Everything in here is going to act as an intron, and recombinase is going to remove it, and we're going to get now everything from V39 to J5 was removed, and they are now together. And you can do that with any combination of these segments. And the book says for this specific example, you have 40 V segments, 5 J segments, and 1 C. You can have 200 different combinations here. And so we're going to get all of this long gene wired down to this all gets removed, and then the intron gets removed. And then what we end up having, you had a cap, poly A tail, and this is how you get your variable region and your constant region for your antigen receptor. And there's going to be, for this example, 200 different combinations. And this is for the light chain. The heavy chain, the book says, is going to have even more possible combinations. That's all for this page, so let's move on to the next one. This part just talks about how recombinase acts randomly. So it's up to chance. Whatever combination ends up in the antibody. Goes on to talk about how after the light chain gene and the heavy chain gene are rearranged, then we can go on to synthesis to the receptor being synthesized. Sorry, I got a stuffy nose. Continuing on with the discussion of combination, this talks about after translation of both the heavy and light chains, they've been rearranged. You get antigen receptors synthesized, each one randomly. For a total population of B cells in a human body, the number of such combinations has been calculated as 3.5, I believe that's 3.5 million. Yes, 3.5 million. So mutations introduced during this recombination add even more variation, making the number of specificities even greater, ergo making adaptive immunity even better at its job. And moving on, we get back to another topic that we had talked about in the first video that I said we would come back to. How does the body distinguish self from non-self? It's a great overarching question of the chapter, and this paragraph on self-tolerance goes even deeper into the subject. Basically, the gist of this section is that lymphocytes that are produced not for epitopes, but epitopes on the organism's own molecule, and then if they're positive for reacting to cells in the organism, they are destroyed by apoptosis. And you guys might remember from class, or if you guys are scientists, know that apoptosis is pretty much cell suicide. The cell really goes pop. That's how I remember it. It's a easy concept. The cell dies, so it's no longer functional. Example, Dr. Nichols gave in class that I find very appeasing is a fetus grows web skin in between the hands, or in between the fingers of the hand. The cells during development that are in that webbing are not needed, so they kill themselves. Since these detrimental antigen receptors kill themselves, the immune system can now exhibit self-tolerance. It will not react or harm itself. Moving on, the next section talks about how, if there are only a small amount of cells specific to one particular epitope of a pathogen, how is any action taken, and it talks about replication of T cells and B cells. They're cloned. Some of these clones become short-lived cells that only take immediate action. These short-lived cells are plasma cells, which secrete the antibodies for B cells. For T cells, they're helper cells and cytotoxic cells, which we will talk about in the next section. There's also a set that remaining cells of the clone become memory cells, which clone more of that needed cells, given if the same antigen is encountered. Clonal selection is simply, it selects what will be given. If you need to pause here to read this, I'm just going to say it. It selects what will be cloned when a certain antigen comes through. Figure 4 through 14, which we'll end on, summarizes clonal selection. B cells generate memory cells and plasma cells. T cells generate memory cells and effector cells. So, on to memory. So, the basis of immunological memory is the basis that we have been discussing about adaptive immunity, where you are presented to a pathogen once, and upon a second presentation, your body is better equipped to fight off that pathogen. So, the first introduction is called you have primary immune response. So, when you are first introduced to a pathogen, or your body is, your response peaks around 2 weeks, so 10 to 17 days, and during the secondary immunity response, if something comes back, your body is more well equipped, and it usually takes about 5 days, so peaking between 2 and 7. So it's more than 2 times as fast the second time around. So this increase in speed, as the book says, is the hallmark of adaptive immunity, or acquired immunity, as it sometimes calls it. Because B cells give rise to the antibodies, you can measure the concentrations in antibodies to distinguish between primary and secondary immune responses. See if you've had something before. It can be used for chickenpox/shingles. I know the book doesn't say that. Well, it does say something about chickenpox, but anyway, it does say that secondary immune response relies on the reservoir of T and B memory cells generated following initial exposure. So the T and B cells have to make long-term protective cells for diseases, as you can see here, for diseases such as chickenpox. And finally here, as we had said, we have figure 43.15, 14 and 15, 43.14 up here, which is a depiction of the cloning selection. You have different antigen specificity, selected B cell proliferates, daughter cells develop into separate categories, memory cells and plasma cells. Plasma cells differentiate, produce antibodies. And down here we have figure 43.15. And down here in figure 43.15, specificity to immunological memory. So up in the graph we have an exposure to an antigen A, and you have the primary immune response here, which has a low concentration, arbitrary units of 10 antibodies, say for, it's arbitrary, so say for 1 molecule of antigen A, you have 10 antibodies. Here it shows a comparative result of a secondary immunity response to antigen A produces to the power of 4 times greater amount of antibodies. Again, it's arbitrary units. It's just a concept visualization that primary response, you have a low concentration. Upon secondary response, you have a much larger concentration. You're infected a lot shorter, and here you're reintroduced to A and introduced to a new antigen B. And antigen B immediately starts, you don't produce any antibodies at first. There has to be differentiation, proliferation, and then you get a primary immune response to B. So that is all for section 2. Next video we will be talking about 3. We'll start here. Adaptive immunity against bodily fluids and bodily cells. Infection of bodily fluids and bodily cells, sorry. And I will see you all then. Thank you for being here, and take care.